Very good. Thank you very much. Well, it's a pleasure and an honor to be back in this room. I think this must be the room where the 3 laureates were first presented. Is that right, or was that somewhere else?
The press conference.
Press conference, yes. And there was somebody in the audience, very aggressive, and said, "Why aren't you working on AIDS?" And all 3 of us said, "What?" And we sort of explained why we weren't working on AIDS. And he seemed to accept it in the end. He became quite friendly, but it was a very surreal kind of experience. So as you can tell, I've been to Korea recently. This is a very beautiful pot. I want to talk about switches in the control of the cell cycle. And you will come to see at the end that I really don't know what I'm talking about. And it will be, I'm afraid, a long journey, ending in ignorance. So here is a switch, and I've always been interested in biological switches. The first switch I sort of studied was the switch which controls the coordination of supply of alpha chains, beta chains, and heme to assemble hemoglobin. And it had been known for a long time that if you block the supply of heme, which you can very easily do by starving cells of iron, that protein synthesis stopped. And it wasn't clear why there should be a connection between heme, which after all, a rather complicated prosthetic group made by a completely different mechanism in a completely different place from the protein, should have any control over protein synthesis. And in the end, it took us about almost 10 years to figure out, and it turned out to be regulated by a protein kinase. And along the way, I also discovered that double-stranded RNA switched off protein synthesis, which if I'd really been clever, I could have probably won a second Nobel Prize for discovering RNAi, but I wasn't that smart, unfortunately. But it was jolly interesting, and this sort of a biochemist's obsession with how things are turned on and off has always been, what interested me. Now, of course, switches can come in various forms, and this is a pretty simple switch, and it doesn't really matter. I mean, at any point you can turn the lights on or off. And the mechanism, what goes on behind this plate, doesn't really necessarily concern the switcher. It might look something like this, for example. This is systems biology, and I challenge you to it actually says at the bottom, it's a radio-controlled light switch. It has to be a lot more complicated than the simple one on the wall. And then there's another kind of element, control element, which I think is what we're going to be really talking about, as you'll see at the end, which is a latch. And I think, if I understand right, I actually came across an electronic engineer that a latch in electronics is a switch with a memory, so it remembers how it was. But in this case, this latch is keeping the gate shut, and the horse that is kept in the field to the right-hand side can't escape because of the action of this latch. But if the latch was opened, then the gate would swing to and fro. So this particular latch is designed to keep the gate shut when it's shut, which is important. If the latch broke, the horse would escape. So here's the view of the control of the cell cycle circa 1961. This is taken from the review on mitosis by Dan Mazier. And the lovely thing is he represented this by a starting pistol. And you can quite clearly see that, I mean, that's not a sort of molecular element or gene product or anything like that. It's an absolutely unknown something that starts things. And you can also see that surrounding this is a pretty complicated set of processes on the left that lead up to the culmination of mitosis, and on the right, the return to interphase. So it's no simple things. And now let's just look at that, what that actually looks like in real life. These 3 red-marked cells, by the way, are the ones which will never divide, but most of the other ones, you can see they just round up and they divide and then they settle back down again. And there's something wrong with these cells because of, I can't even now remember what this experiment was designed to investigate, but I think they've been injected with something that messed up their division. But I think you can see that the different cells, even though they're synchronized, they divide at different times. And it is my contention that actually we still, to this day, do not know what it is that actually triggers that rounding up, the entry of cells into mitosis. If you were God, you would probably try and design the circuit so that the cell was aware of how much DNA it had synthesized, and when it had completely finished synthesizing DNA, then and only then would the cell be allowed to divide. So that, but then if you think about that, it would mean that there has to be some incredibly sensitive sensing device that can detect just one replication fork in the whole cell. And I understand from my friends who work on replication that actually the evidence disfavors that idea. Although it's true that if you inhibit DNA synthesis in most cells, the cells will never divide, or if you damage them heavily enough, they will never divide. But there's really no evidence that just one little replication fork can delay the entry of cells into mitosis, which then raises the question of how they know when they should actually enter mitosis. And you know, this is something that I think it would be nice to work on. I mean, I've sort of been trying to work on that rather ineffectually for the last 20 or 30 years. And it's very nice, actually, I think, when there are still things we don't know, because it means that those of you who still have your careers in front of you have some good problems to work on. And finding good problems to work on is no easy matter, let me assure you. The hardest thing in all of biology. Anyway, this is not terribly dramatic. I mean, you can see that the cells are divided. You can't see what's going on inside. If we look a bit inside with a friend who labeled the chromosomes with a fluorescent dye, you can here see them. Now they've finished division. There are twice as many cells on the plate as there were before. Now, you can't really see it, but they are actually synthesizing DNA. And eventually, I think they should come back again. Yes, here we go. We've been through it. It's very speeded up, this movie. It's 24 hours in about 24 seconds. And that's the whole point of mitosis, really, the division of the chromosomes. So leading up to cell division, things sort of happen slowly, but there are some quantized events like this pole splits into 2, then the chromosomes start to condense. We still, by the way, don't really understand the basis of chromosome condensation. Some of the enzymes are known, but what they actually catalyze, what they actually do, is really not known. Terribly important, they should condense, because if you think about it, if they didn't condense, the way that this works at the height of mitosis is that these little chromosomes are pulled apart by kinetochores, which are protein DNA adapters, which are pulled by motors that work on these microtubules and are pulled apart here at the metaphase-anaphase transition. Now, if the chromosomes were not condensed and they still you'd pull, but it would be like sort of, you imagine a tangle of wool, and you just pull a bit of the wool towards one pole, and the rest of it would just stay in the middle of the cell somewhere. So it would be an absolute disaster. Tremendously important that the chromosomes condense down to a smaller size, actually, than the dimensions of the cell. And as I say, we have absolutely no idea, really, of the basis of that condensation. It's still quite controversial. And then the cell waits a good time, and we'll come back to that, and doesn't divide until the 2 sets of chromosomes are well apart from one another. And then and only then does a sort of little circular muscle divide the cell in 2. And this is a complicated process which is finally beginning to yield to investigation by some very clever people. And again, it's quite a surprisingly complicated process. So a lot is going on. And the question is what controls it, and the answer is, to cut a long story short, that there is an enzyme, and this is it, with is actually CDK2 on the left and cyclin A on the right. And it's this thing, this enzyme here turning on, which brings about the entry of cells into mitosis. And the question is, how does it do it? And the answer is that the business end is the protein kinase on the left, and the cyclin, the protein that I discovered on the right, which is necessary but not sufficient to turn it on. And work in Paul Ness's lab clearly revealed the existence of a control mechanism whereby, interesting, an unusual protein tyrosine kinase called WEE1 places an inhibitory mark on the CDC2, and so that even if the CDC2 combines with its activator, it's still not active, and the inhibitory mark has to be removed by a protein phosphatase called CDC25. So if you get rid of WEE1, what it means is that CDC2 becomes prematurely active, and the result of that in yeast is that the yeast divide at a smaller than usual size. It accelerates entry into mitosis. And then this enzyme catalyzes the interphase to mitosis transition. Now, there is a problem with this WEE1 control, right? WEE1 inhibits CDC2, but it turned out that actually that is true in interphase before you enter into mitosis, but once you get into mitosis, CDK1 inhibits WEE1. And I think you can see that this is a sort of typical kind of switch that you often find in transcription factor circuits when a cell can either be A or B, but it can't be both. And so you set up circuits of this kind where one gene product inhibits the transcription of the other gene product. So whoever, in that case, turns on first wins, and you can't go back the other way. But we have a problem here because in interphase, WEE1 is keeping CDK1 in check. And the question is, how can you ever get out of that? And the answer is, well, it's done by CDC25, the phosphatase that antagonizes, but we don't know how CDC25 is activated on time, or what controls its activation. Once the switch has been thrown, however, it's stably in the other form, because once this CDK1 turns on, WEE1 is held in check. And the way that we then get back to the interphase state is by degradation of the cyclin subunit so that the protein kinase is no longer active. So that's easy. But when you look at this, you say, well, you know, it doesn't look like, it really looks like the cell cycle has two stable states, which we can call mitosis and interphase, but you could never get from interphase to mitosis or mitosis back into interphase. So it's problematical. But basically, the story is a very simple one. The cell, there's always CDK1, the kinase subunit there. And what happens when things turn on is you just start accumulating the activator. At some point, you have to assume that the activator goes through some threshold, and the CDK1 turns on, turns off the WEE1, and the cell enters mitosis. And then the cell degrades cyclin, and CDK1 turns off, and the cell exits mitosis, and that's what you read in the textbooks. It's really simple once you know what's going on. It took a long time to figure that out because people didn't know very much about protein kinases, and people never dreamt that proteolysis, the specific degradation of a protein, could occur inside cells. I mean, the only kind of proteases which were really known at the time were the kind that you have in your stomach, which is the kind that I find my kids still being taught about. When it comes to enzymes in England, enzymes mean proteases, things you digest your food with. And that's about it. You know, enzymes don't do anything else. You know, you can't see with an enzyme. And actually, I was very entranced by the idea that an enzyme could catalyze a cell cycle transition. I thought that was fantastic. But then you begin to think, if you're a biochemist, then you begin to think, well, hang on a moment. This is an enzyme. It sticks phosphates on other proteins. So exactly how many proteins does it have to phosphorylate by how much in order to bring about this unfolding series of events that we call entry into mitosis passage through mitosis, the metaphase-anaphase transition, and back out into interphase the other side. And it turns out this is not such an easy problem to solve. One would like to have a sort of a complete catalogue of all the proteins that are phosphorylated and to have a little inventory of what the phosphorylation does to those proteins in order to make the cell undergo this amazing reorganization. I mean, you know, how do you break down the nuclear envelope? How do you assemble the mitotic spindle from the interphase array of microtubules? And actually we have a surprisingly poor understanding of that. But basically, it's fairly simple. I mean, you know, here are a bunch of substrates which don't have much phosphate on them. You turn on the enzyme, they all get phosphorylated, and this state somehow is mitosis. Some of them get multiply phosphorylated, as you'll see. And inventories that have been performed reveal that actually there are hundreds of substrates, many with these multiple phosphorylation sites, that get phosphorylated when the cell enters mitosis. And then something that is a little bit more worrying, you say, well, you turn off the protein kinase and of course you come out of mitosis, but of course you only come out of mitosis if it's true that it was sticking those phosphates on that got you into mitosis, then presumably it's true that taking those phosphates off is what gets you out of mitosis. So there's an element in here that we haven't considered at all, namely the enzyme that catalyzes this dephosphorylation. And historically speaking, two approaches to this have been made. Geneticists isolated mutants that were stuck in metaphase and couldn't get out of mitosis. And it turned out that almost all the mutations mapped in an enzyme called protein phosphatase 1. But when biochemists looked at the problem and took typical mitotic phosphate substrates like this and assayed what took these phosphates off, they said that it was actually a different enzyme in the same family called PP2A that took them off. And I actually witnessed almost fistfights between the geneticists and the biochemists who said it's PP1, no, it's PP2A. And after a while, it got kind of boring because they never seemed to be able to reconcile their findings. And eventually it just went quiet because it was so unedifying that it didn't seem much use, really. So people sort of forgot about the problem. But if you think about the problem, and this is something that one learned in biochemistry and very early on, was that we have here a typical example of a futile cycle. If we have the kinase on, we take a substrate with a hydroxyl group, ATP is hydrolyzed, the terminal phosphate of the ATP is transferred to the substrate. And if the phosphatase at the same time is active, of course that phosphate comes straight off. Two consequences. One, we burn a lot of ATP for no reason at all. The cycle just goes round and round. And number two, we can never get fully phosphorylated because the more of this stuff that accumulates, the faster this guy will work, just by mass action. And in fact, one of the things that I came across was a case where the kinase was regulated and the phosphatase was not. And in that situation, you never got more than about 30% of the substrate phosphorylate. This was, in fact, the heme-controlled situation, and we were very puzzled at first how only 30% phosphorylation of the target could give you virtually 100% inhibition. It turned out that the reason was because the phosphorylated form was a kind of dominant negative inhibitor. It actually clogged the works and stopped another little cycle from working. But that was a case where this futile cycle was acceptable in the name of control, but somehow they didn't seem very likely, and you would have thought that for the cell cycle, which has these two very different states, interphase and mitosis, that it's awfully likely that when the kinase is on, you would turn the phosphatase off. I mean, this is terribly simple-minded stuff, and if you simply think of trying to fill a wash basin with water, it's bloody obvious that it is true you have to turn the tap on. There has to be a source of water, otherwise the sink will never fill. But to fill up the sink, you have to put the plug hole in. And if you think of the tap as the kinase and the plug as the phosphate, I can't see how this one is regulated, actually. But there's usually a little thing these days behind the tap. But sometimes you just have to press it. It goes down. I was brought up with ones which were on a chain and were made of rubber, you know, and you stuck them in the hole. So this is, you know, this is real systems biology. And And the hero of the story is a Japanese postdoc who came from my friend Mitsuhiro Yanagida's laboratory in Kyoto, and his name is Satoru. And he is the person who will do all the next series of experiments. And you will see that he was a terrifically good experimentalist. And he was my last postdoc, and it was really a pleasure working with him. I cannot tell you, I mean, one of the most satisfying things about being a scientist is working with other people. And it doesn't terribly often happen that you work with somebody else who you are on absolutely the same wavelength with. And it turns out it doesn't really matter whether there's an age difference between you, but the fact is, if you can tell somebody, no, Satoru, that's a stupid idea, and he can tell you back, no, Tim, that's ridiculous, don't even think about trying to do that. This is really great, because science is something where you have to be very blunt and honest with each other, and if you take offense at that kind of honesty, you're in trouble, because the truth is often a little bit painful and hard to accept. Anyway, I mean, I can't tell you how sorry I was when Satoru had to go back to Japan, where he now is. So the system that I'm going to be using from now on is this one, which was originally invented by Fred Locher and when he was a postdoc with Missouri, actually, in Toronto. And then he went and worked with Jim Mahler, and they succeeded in purifying what was then known as MPF, maturation promoting factor, which later turned out to be mostly CDC2 and cyclin B. And then Andrew Murray refined, and another Japanese worker refined it further. And basically what you do is you take frog eggs, which are arrested in meiotic metaphase 2. So they're there's not actually mitosis, but it's very close to it, and they're deeply arrested, as we'll see in a second. And you spin them 10,000 revs per minute for 10 minutes or something like that. And actually this is the second spin, and by spinning them, there's a lot of rubbish, yolky stuff that goes to the bottom, a lot of rubbish, lipidy stuff that floats to the top. But in between the rubbish at the bottom and the rubbish at the top, which is probably very important, and one shouldn't be rubbishing the rubbish, but in between there's this wonderful yellow layer of cytoplasm. And if you take this wonderful yellow layer of cytoplasm, which is sort of metabolically active, and you add nuclei to them, which you've demembranated, this is what it looks like. Absolutely amazing. This cytoplasm is in M phase, and it knows how to assemble chromosomes on gorgeous, typical second meiotic spindles. I say they're typical second meiotic spindles because you'll see usually if it was a mitotic spindle, there'd be microtubules coming off at the poles, but because they're meiotic, sort of people talking about be barrel-shaped, but all the microtubules will point inwards, and there are the chromosomes, there are the two poles, and it's fantastic. And now, if you were to add just a little dab of calcium, the chromosomes would come apart. and so it's amazing. I mean, this is a completely cell-free cell division system. It's fabulous. And it's very attractive. Now, the only trouble is, of course, it's completely, I mean, the one thing you don't have to do, therefore, is any micro-injection. You can just use an Eppendorf pipette to add things, or you could add antibodies on bees to subtract things, and we're going to do quite a lot of that from now on. Now, one other thing I need to explain. So here is the unfertilized egg. It naturally has its real meiotic spindle. So this has gone through it's gone through one meiotic division, meiosis 1, separating the homologs, and now it's about to do meiosis 2 to separate the sisters. A sperm would normally arrive at this juncture, and you'd get a male pronucleus which would come and fuse with the female pronucleus, and then you go into a series of mitotic divisions. And what genius Missouri discovered was that if you suck out the cytoplasm, this is before the centrifugation, which would have made life a lot easier, with a fine needle and inject it into another oocyte, and the oocyte is arrested in G2 of the cell cycle, this stuff, which he called MPF over this side, made the oocyte behave as though it had seen progesterone, and it would turn into an egg. So there's something positive in here. But if you do the other thing and ask what happens if you take this same cytoplasm and inject it into a fertilized egg, the first round of DNA synthesis, nuclear fusion goes beautifully, but when the cell reaches metaphase, or first mitosis now, it stops. So this has got both a promoting activity and a stopping activity, and the stopping activity is called cytostatic factor, CSF. The MPF is the active stuff that triggers the G2 to M transition, and CSF is the stuff that holds you, should be holding you in meiosis II, but can also perfectly well hold you in mitosis at any stage. Okay, so what is CSF and how does it work? Well, it took a long time, as you can see here. The first surprise was that CSF was apparently an oncogene called CMOS, and then Angel Nebreda in my lab and John Cooper in Seattle discovered that MOS activated a MAP kinase cascade. And then some years later, Jim Farrell and Jim Mahler found that actually the end of the cascade was an enzyme called P90 RISC, which was required. And then Peter Jackson in 2001 identified something he called EMI1, which was really CSF and presumably downstream of all this signaling pathway. And then there is a little gap here, another 4 years, because it was discovered that actually the eggs don't have any EMI1. So it was extremely unlikely that something which isn't there could be the regulator. And we had a bit of a tussle over that, actually. And what was really irritating, because actually there's a Cell paper and a Nature paper which says that EMI1 is CSF. And they're both completely wrong for a really trivial, I mean, something that I would almost fail an undergraduate for doing. I mean, they're based on an antibody artifact. The band they thought was EMI1 was nothing of the kind. It was a cross-reacting band at roughly the same molecular weight. But Thomas Meyer then discovered that ERP1 was the real inhibitor of the metaphase-anaphase transition. And irritatingly, ERP1, this stands for ME1-related protein 1. And Peter Jackson was basically right, although he was actually wrong. I'll show you in a second so you can get a sort of pretty much sense. So how does this all work? Well, it turns out that you need P90 RISC to activate ERP1. So the whole thing, the MOS is upstream. That, by the way, is synthesized during the early maturation reaction, and everything works much better if you have some MOS. Activates P90 RISC, activates this, which inhibits the enzyme that degrades the cyclin. And the APC, anaphase promoting complex or cyclosome, is a ubiquitin ligase, which is very specific for a certain set of proteins, including cyclins A and cyclin B. Okay, so that's all very fine, but now we're arrested, right? Because we've got this inhibitor of cyclin degradation, so we stay in M phase forever, unless we can get rid of this inhibitor. So how do you get rid of this inhibitor? Again, it's calcium, you know? We were discussing this earlier, you see. So how does the calcium work? Well, it's absolutely amazing. It's a really baroque mechanism. So CAM kinase 2, I think this is an enzyme which is supposed to help you remember things, if I remember all right, phosphorylates ERP1. That doesn't actually do anything in itself, but it generates a site for polokinase, which phosphorylates phospho-ERP1. Then that phosphate is recognized by a ubiquitin ligase called beta-trip. I don't know how you pronounce that. And that leads to the degradation of the inhibitor, and that means that you release the inhibition of the APC, cyclin is degraded, and off you go. I mean, really, it took a long time to work all that out. And here is ERP1 itself, and it has a business end which interacts with the APC. And when Satoru joined the lab, the experiment was to find out how this worked. And the reason why I thought that was a good project was because of a lunch in a Chinese restaurant in San Francisco with an old friend called Tim Mitchison. And Tim told me that this stuff, GST-ERP1, was a fabulous inhibitor of the mitotic arrest of the system, which Tim needed because he was working on microtubules, and it's the best inhibitor that money can buy. So I suggested to Satoru to make it, and then we find out how this stuff actually did the inhibition. It was supposed to be some kind of a pseudo-substrate, but we were a little bit puzzled, and this is something too which has never been really fully resolved. We found that there is about 300 nanomolar APC in this extract, and it only took 28 nanomolar inhibitor to inhibit it, which doesn't make sense because it's supposed to be a stoichiometric inhibitor, but actually it seems to work way substoichiometric, sort of implying it's enzymic, or only a very small fraction of the APC is ever active. Don't understand that. Anyway, the important thing is really not that. It's that Satoru succeeded in making this stuff and in testing it. And now this is where the assay system comes, because you're going to see some more of these things. So this is just a marker for mitosis. It's called APC3. It is actually a component of the APC. It gets multiply phosphorylated, so it shifts up in mitosis. So that's mitosis. This is interphase, and you can see that in, oh God. Who is this? I'm giving a lecture. Hello? I don't think she's there. I think her handbag is ringing me. That's good. That was my wife. Had to answer. Okay, up in mitosis, down. So you can see that there's a phosphatase is turning on here, right? Clearly. The important bit really is this. In the control, you add calcium, there is a delay, and then the cyclin disappears. Right? If you add this construct here, you add the calcium and the cyclin doesn't disappear. So Satoru came to me with a big grin on his face saying, it worked. I say, great, but what's happening here? He said, I don't know, but it always happens. So I said, well, you know, you've just added calcium, so it's obvious what must be happening. You can see this thing is getting transiently dephosphorylated, and because the cyclin isn't going away, the kinase stays on, so we have a contest. A phosphatase, when the calcium levels are high, is winning here, but it never fully succeeds, not like it does here, and then it goes back. So there must be a calcium-activated phosphatase in the system. And he agreed, and we set out to find out what it was. And it wasn't very long before we found out what it was, because it was obvious what it had to be, really, and just a matter of proving that was. This is hypothesis-driven research, I guess you could say. So there's an enzyme called calcineurin. It gets its name because it was a calcium-binding protein found in the brain. It was many years before it was discovered, I think by Philip Cohen, that it was actually a protein phosphatase. And it is known to be the target of an immunosuppressive drug called cyclosporin A. In fact, this enzyme is the basis for all kidney and heart transplants. And rather inhibiting it is, because if you inhibit it, T cells can't develop, and therefore you get no T cell immunity when you take the drug, and the kidneys don't get rejected. So this is very handy because it means you can easily test. So Satoru looked up and got the assay for calcineurin. Sure enough, there's no calcineurin activity in the egg system until you add calcium. Then you get a burst of activity, which very rapidly declines because the calcium that you add is very rapidly sucked up into mitochondria and other vesicular structures that are present in this crude extract. So it's transient, and you can abolish it by adding this immunosuppressive drug that you just buy from Sigma. So now we're in a very good position to ask whether this is important for the metaphase to anaphase transition. So do the following experiment. We take demembranated sperm nuclei and add them into the yellow cytoplasm. In one tube, we don't add anything. In the next tube, we add calcium, which will return it to interphase. And in the third tube, we add calcium plus the immunosuppressive drug to inhibit the calcineurin. And you can see here that whereas the nuclei look like this when you have these funny shapes, if all goes well and we return to interphase, then the chromatin inside the nuclei swell. They form a nuclear envelope, and the nuclear envelope, this middle thing, is sucking up a fluorescent nuclear localization signal tagged peptide. So the nuclei are intact and they're working. And you can see that if you If you don't add anything, then the chromatin just doesn't swell, it just stays the way it is. And if you add cyclosporin A to inhibit the calcineurin, the swelling of the chromatin is very inefficient, and you don't form nuclei. And of course, because there are no nuclei, they can't suck up the nuclear protein. This is the actual assay that's plotted over here. And you can see that adding the calcineurin inhibitor delays the return to interphase by roughly an hour. So it's really important, this phosphatase. So this sort of really gets us thinking hard about the role of phosphatases in getting out of mitosis. But we're still thinking calcium at this point, and it has been suggested that calcium is important for getting out of mitosis. So we find that adding calcium later on, so it's very important for getting out of meiosis, meiosis 2, but in the mitotic divisions that follow, adding that drug has no effect whatsoever. So I breathe a big sigh of relief because calcium isn't important after all for cell cycle transitions. But then we start worrying about it, and we worry because what the textbooks seem to imply is that there is a sort of background level of phosphatase activity, so that's what gets you out of mitosis. And every so often, this is sort of overwhelmed by an enormous, irresistible spike of kinase activity, and that's what gives rise to this pattern. But if you actually measure the kinase activity, you find that it really goes up rather slowly in most of our experiments, and it kind of follows the cyclin B levels. In other words, the kinase activity does something more like this. And in order to explain this abrupt switch-like behavior, you really have to say that normally there's a phosphatase which is always antagonizing the kinase, which is somehow then switched off, stays off for mitosis, and comes back on again after the cyclin has been degraded to get you out of mitosis. So, what does this mean? It means that we have to start assaying phosphatases. And assaying phosphatases actually is much more easily said than done. Because to assay a phosphatase, you need to have a substrate. So after a couple of false starts, we decided that this was going to be our assay system. When I say we, of course, I mean Satoru. So what he did was to fuse, using standard molecular biology, maltose binding protein, a nice well-expressed protein in bacteria that you can purify on a simple affinity column, and stick 25 residues surrounding serine 50 of an enzyme called CDC20, which is actually a co-activator of the APC, and that's sort of irrelevant, really. We knew that this serine, which is a classical MPF site, S, P, something, K or R, gets phosphorylated in M phase and dephosphorylated in interphase. So it was a good substrate. And despite the existence of all these other serines which could get phosphate, it turned out that none of them did. So he made an alanine mutant that doesn't get phosphorylated. Then you take this stuff out of the bacteria, phosphorylate it with cyclin A, CDK2, and gamma P32 ATP, and you get this labeled substrate, which you can then add into the crude extract and ask how much activity is there by just incubating for a couple of minutes and asking how much phosphate comes off. Very, very, very simple. I mean, it's slightly tricky. You'd sort of wish it was a bit easier than this, because you see there's a lot of stuff you have to go through first to get the substrate, and it uses radioactivity. So the first shock we got, or a shock, I mean, it was really rather gratifying, was that in that meiotic extract, there is no activity against that substrate whatsoever. It's completely stable. Yet if you take a mitotic extract, in interphase, there is good activity against that substrate. Gosh. And then you get this very funny result, which is that when you add the calcium, it goes up and then it goes down. If it had been anybody else, I would have said, do the experiment again. You obviously dropped that tube on the floor. But it wasn't. It was Satoru, and this is absolutely the correct result. And when we started exploring it, we found by use of inhibitors that the first peak is susceptible to inhibition by calcineurin, and the second peak is inhibited by okadaic acid, which is an inhibitor of both PP1 and PP2A. So we're dealing not with one phosphatase to get you out of mitosis, but at least two phosphatases, and possibly more, because I say okadaic acid is really rather a blunt weapon. So it looks like what's going on here is that actually you need this first phosphatase peak to turn on the second phosphatase peak. Gosh, so what is the second phosphatase peak? Well, but we don't know what it is, so we called it phosphatase X. And the first really cool experiment, which is sort of almost too good to be true because we didn't know any way of identifying phosphatase X or of working out how it's turned on. You just ask what are its properties, and much to our delight, we found that in a mitotic cycle, so this is now, we add calcium back here and get cells to synthesize cyclin B, which makes them enter mitosis here. Sure enough, phosphatase X, whatever it is high in interphase and gets turned off completely when the cells enter mitosis. Terrific. Almost too terrific. I don't like it when you get such clear-cut results because it makes you think that somebody might have forged them or something. But it turns out that other people agree too, so I'm sort of relieved by that. So, what is the enzyme and how is it turned off? Well, the first clue, it's no easy matter to do this, actually. So again, being sort of, I always like, if I'm up against a brick wall, I like to try and find a secret way around it. So this secret way round was just to ask a simple question, is it possible that it's just CDK1 that is regulating the phosphatase? And the answer was, to a first approximation, yes. And this experiment just adds in a very specific inhibitor of CDK1. It's actually a protein called P27. And you titrate it in, and as you turn off the kinase activity of CDK1, phosphatase X comes on. So we can kind of draw a circuit like this, which says CDK inhibits. And we don't suppose that it's quite as simple as that. There's probably stuff in between, as you'll see there is. But that's the logic of the circuit. So we go back, remember the futile cycle diagram. This is exactly what you want to get rid of the futile cycle. So now if we inhibit phosphatase X, of course, it's very easy. This now catalyzes, you can get full phosphorylation of these proteins. Terrific. Everything looks wonderful. So we decided to isolate phosphatase X. So how, well, biochemists, remember, so you just purify phosphatase X and find out what it is. But how do you know it's phosphatase X? Well, the reason is that it's off in M phase and on in interphase. So what you do is, and to do that you have to run columns and do ammonium sulfate precipitations and typical enzymic-y things. And we got a shock when that happened because we found if we took this mitotic extract, which has no activity of phosphatase X, and simply diluted it with buffer, the phosphatase activity came on. So much so that by the time you diluted it to the point when you'd actually be using it to run on columns, there was essentially no difference between the two extracts. Bad news. So we thought about some things which might possibly be important for doing that kind of regulation. We thought, well, maybe it regulates itself by phosphorylation. So we dilute the extract, the ATP concentration goes down, the thing turns itself off perhaps, something like that. So we try adding ATP to the buffer. It doesn't do anything really. We think, well, it's very dilute protein. Maybe the thing's a bit floppy and it doesn't like being in likes to be in concentrated protein extract, so we'll add some BSA, that helps a little bit, and the two together help a little, but not really. So finally, but it took about 2 years, I persuaded Satoru to do my favorite experiment, which is called the boiled control. If you're in a situation like this, the obvious explanation for this behavior is that there's an inhibitor in here, and the question is the inhibitor large or small? If it's small, if it's sort of something like, manganese ions or coenzyme A or something like that, you can boil it and it's still active. So I got Satoru to boil the extract, and it mostly turns, of course, into hard-boiled eggs. There's a tremendous precipitate form. You spin off the precipitate, get the supernatant, and do the dilution in the supernatant, which contains all the small things, the heat-stable things. And sure enough, if you do that, you don't get this turn-on phenomenon. So there is something in there which is heat stable, there is a target. Now that took a very long time to come around to, so I'm sort of getting in my head, but it'll help explain something when we get to it. Meanwhile, I asked all the phosphatase people, what the hell is going on here? This looks very odd. And they said, oh, well, you know, we've always known if we wanted to purify, get a good yield of phosphatase from rabbit muscle, you must use a large volume of buffer. I said, why? And they said, we don't really know. So they weren't very helpful. I should say, however, that the phosphatase people were in general. I mean, one of the things I learned along this whole story, we were constantly getting reagents and advice from people because we really had no experience. And the people in the phosphatase field were fantastically generous, sending us constructs and help. You know, there was one enzyme you could only purify by including ascorbic acid in the buffers and things like that, which wasn't described in the literature. It was really extremely heartwarming. You know, you'd explain why you wanted to do something, they'd come right back and, yeah, here you are. It was great. So I've said, I just include this to make sort of ram home a point that there is this drug called okadaic acid which inhibits most of the, but not all of the phosphatases. You've seen it doesn't inhibit the calcium-activated phosphatases. And actually, if you take one of these extracts that hasn't synthesized very much cyclin, the only thing you have to do to get it to enter mitosis is to turn off the phosphatases. You can do that just by adding this blunt weapon, okadaic acid. So, and you can see, another thing we introduce here is the idea that you can buy an antibody from New England Biolabs that reacts with SPs and TPs and shows you a whole multitude of phosphorylated proteins as the extract enters mitosis. And you can see here APC3 shifting up tremendously when you add okadaic acid. So there's only a little bit of cyclin here. So it's really more important to turn off the phosphatases probably than to turn on the kinases. It's a sort of good shortcut. It won't do you any good because it kills the cells pretty quickly. It's not a very useful drug actually. In fact, I think it's the basis of, you know, if you get shellfish poisoning, this is what kills you or makes you very, very sick. Anyway, here's the family which we're probably looking at because it clearly is this family, the okadaic acid sensitive family. PP2A, and the major ones are PP1 and PP2A. And this is calcineurin, which is not susceptible to okadaic acid. Okay, so to cut a very long story short, by means of a long, laborious set of immunodepletion and phosphatase assay experiments, we discovered that the enzyme we were looking for is a form of PP2A with this B55 subunit on it. And this is what the structure looks like. And that was helpful because the people who did this structure, of course, had all the components for making and the instructions for making that phosphatase. And they very kindly sent us all those constructs, which will come back and be useful. So there's a sort of a scaffolding subunit which binds this catalytic subunit and what is called in literature a regulatory subunit. And it's not really clear quite what the regulatory subunit does. I mean, presumably it plays some role in substrate selection. So how might it be regulated? Well, we noticed that the B subunit, that's this guy here, has a juicy mitotic phosphorylation site in it, which is quite well conserved in evolution. So for quite some time, we tried to show that this site was phosphorylated, and you could get some label into it, but never very much, and it didn't look very promising. So it looked like the real answer was probably more something along these lines. PP1 has long been known to be inhibited by something called inhibitor 1 or DARP32 and various other names that and it's a phosphorylatable phosphatase inhibitor. It's a kind of a pseudo-substrate which, when phosphorylated, sticks in the active site but can't be dephosphorylated because of some other feature of the structure. Maybe it's held just out of range of the active center or something, and presumably has some sort of binding affinity with the selector subunit. So now once that thing is in there, a real substrate can't get access to the active center of the enzyme. So we looked for that. We knew when the enzyme was inhibited. We had antibodies against these subunits. We pulled it down and we looked for things like that and miserably failed to find them. So we began to rather despair of that as the right answer. And then we began to read this gentleman's papers. And this is really the key. This is Mike Goldberg, and this is his first paper, Great Wall Kinase. You wouldn't have noticed a nuclear protein required for proper chromosome condensation and mitotic progression in Drosophila, 2004. Why is it called Great Wall? Simply because the graduate student who worked on it was Chinese. So I must admit that I missed this paper when it came out. It didn't ring any bells at all, but when this one came out in Molecular Cell, we perked up a good deal. Great Wall Kinase, same enzyme, participates in the CDC2 autoregulatory loop in Xenopus egg extracts. Now we're on home territory. And it quickly became apparent. I went to a meeting in France and saw a poster by Anna Castro, who is here, and the poster made a very compelling claim that actually the way that Great Wall worked was by turning off PP2A. Ta-da! And in fact, and then when this, after this was published in the EMBO journal, Mike, who became a good friend in the course of this, asked us for antibodies, and so Satoru's name is on the paper, and they showed actually the form of PP2A that's inhibited was our friend PP2A B55, and not one of the other forms. So then the question was, you know, what does Great Wall is a protein kinase, so it must do whatever it does by phosphorylating something. So probably it ought to be phosphorylating that hypothetical phosphatase inhibitor. So Satoru decided to do something which, you know, just completely unbiased actually. And what he did was to learn how to make Great Wall, thanks to constructs sent by Mike, and who had been looking for this and been doing peptide arrays and getting no luck whatsoever. And when we took the boiled extract, now why did we take the boiled extract? Well, because we knew that what was supposed to be inhibiting was in the boiled extract, and because boiling the extract gets rid of the background. If you just take a cell extract and you add gamma P32 ATP to it, you will see everything black as hell. But as you can see here, if you add it to the boiled extract, there is very little phosphorylation of anything. And likewise, if you take the Great Wall thing, there's very little phosphorylation of anything. But you mix the two together and Great Wall sees something in the boiled extract that it absolutely loves. Fantastic. So what do you do now? Well, you go up the hall to visit this machine and these people, and you say, can you tell us what this band is, please? And they say, sure. And they're very nice about it. But they come back with this long, meaningless list. And you look at this list and you say, oh, God. You know, they're all about the right size. This band is about sort of 19, 20 kilodaltons. So everything, of course, has the right size. But you would never have guessed that it was MGC 79021 that you were after. So what do? Well, if you're Satoru, here's what you do. You take all the likely candidates and you clone them and express them in E. coli. Here they are. Doop, boop, 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 boop. It looks like a sort of modern music score. I like to think of it as that. And then you mix the ones that are heat stable, because you know it has to be heat stable, with Great Wall, which you've by this time learned how to make. And sure enough, you know, lane, whatever it is in lane 25 is the stuff. In fact, it's amazingly much better than the synthetic substrate that everybody's been using up to now, which is myelin basic protein. Really not a very good substrate at all, or maybe we got the wrong batch of myelin. But anyway, this is terrific. And what is it? Well, it turns out to have been discovered by Paul Greengard. And there are only about sort of 5 papers on it in the entire literature, which is a wonderful thing to have happen. Because normally when you feed in your favorite protein, you find there are sort of 10,000 papers. And reading 10,000 papers is, well, it's just impossible. You don't know where you are. And it also turned out to correspond to something called alpha endosulfine, which was supposed to explain insulin metabolism. But unfortunately, it didn't really, and that sort of went away. But there are 2 of these things. And again, simply reading the literature was so small that we came across this paper, Alpha endosulfine, they're very similar, these two proteins. They're a conserved protein required for oocyte meiotic maturation. So the knockout's been done, and you know you've got, this gives you tremendous confidence because it has the right phenotype. But these guys are geneticists, and I'm a biochemist. And the authors of this paper were very puzzled because they couldn't understand why these oocytes weren't entering M phase, because they had high CDK1 activity. And high CDK1 activity, as you know, means you're in M phase. But being geneticists, it would never occur to them that there might be something that was opposing metaphase in the form of a phosphatase. And when I saw this, it was sort of obvious, seeing through the eyes of a biochemist, that they had indeed stumbled upon a phosphatase inhibitor. So then we found out where the phosphate was put on these two. This is showing the conservation between ARP and endosulfine. And it's this phosphate here. It's a rather unusual sequence, and it doesn't look like anything else really in the database. And it's a sort of conserved little peptide that you only find in this family of proteins. So then the question is are we actually right? And this is it looks so boring, but this is the experiment, because what you do is you take the catalytic monomer and you add either no Ensa or Ensa with a hydroxyl group, i.e. not phosphorylated, or Ensa phosphate, no effect. Put together the A subunit, the scaffolding subunit, with no effect. But stick on the B subunit and suddenly the holoenzyme dies. So we are home and dry. That's the answer. This is a phosphorylatable phosphatase inhibitor that is specific for this form of PP2A. And something I should have mentioned en passant is that this form of PP2A in the frog egg extract is about 30% of the total. 70% of the PP2A is associated with B prime, B double prime, and B triple prime subunits, and they do not bind to this inhibitor. They do not get inhibited by this inhibitor. So it's very specific for this particular form of PP2A. But most people, when they talk about PP2A, do not distinguish between these different forms, and we really don't know. I thought there would be a huge literature which would tell you exactly what B55 liked to dephosphorylate in comparison with B56. You'd think there'd be lots of papers in the JBC, and there are not. It's very surprising. And it just shows how much the phosphatase field lags behind the kinase field. So then you've got to do some more experiments. First of all, this is a stoichiometric inhibitor, right? And I'm glad to say that there was about twice as much endosulfine as there was of the B subunit. So there's enough in there to inhibit. Does it get phosphorylated when the cells enter mitosis? Yes, it does. So here is endosulfine run on a gel which emphasizes its phosphorylation, and you can see that essentially very little is I wouldn't say none is phosphorylated during interphase, but as you come into mitosis, it all gets phosphorylated. Terrific. And Great Wall is running, I mean, almost everything is, you can see. In fact, this experiment was really designed to see which came first. Was it that the activity of Great Wall precipitated entry into mitosis, or was it that this was a downstream consequence of entering mitosis? And the answer is you can't tell, because everything happens at the same time. And then we asked what happens if you take away the endosulfine, and the answer is that when you take away the endosulfine, here's the control going through, this is a very beautiful experiment, the control goes through 2 cycles, cyclin gets synthesized, enter mitosis, cyclin is degraded, out of mitosis, cyclin is remade, enter mitosis again, cyclin is degraded, out of mitosis again. If you take away the endosulfine, so you can no longer inhibit the phosphatase, the result is that you turn on the kinase, right? There's the kinase activity going up. Cyclin is, but you never trigger its destruction, and you never really fully phosphorylate APC. Compare the intensity of that band there with that band there, right? It never shifts up. And if you look at these phosphoproteins, which go very dark in these mitotic lanes here, it's sort of very pale here. So you just can't. So, okay, we've obviously got it right. This is the answer, and This is the enzyme that opposes entry into mitosis and presumably gets you out of mitosis. So we then turn to test that hypothesis, and I'm warning you, this is where things begin to get puzzling, and we are in for a nasty surprise. Okay, first experiment is a triumphant success. What would you expect would happen if you can't turn off the phosphatase? Well, if you can turn off the phosphatase. So here, actually, the experiment is to remove PP2A B55. So the phosphatase is just gone. So now there's nothing to oppose the kinase, so the result is that you should find it much easier and quicker to get into mitosis, and you do. Here in the control, we enter mitosis after an hour. Here in the depleted extract, where there's no phosphatase, it's only half an hour. And you do so at sort of half the cyclin concentration, so it's much easier. And once we're in, we stay in forever because we've got rid of the enzyme that gets you out of mitosis. Obviously, we can't get out of mitosis now because we've got rid of the enzyme that gets you out of mitosis. So really pleased with ourselves, we submitted this to the EMBO journal, and thank God one of the reviewers said, pretty nice, but we'd like you to do just one more experiment, please. And thank God they did, because the one more experiment, you remember earlier on I used this P27 trick, so this doesn't rely on cyclin degradation. And actually, if you look here, it's slightly embarrassing because the cyclin never really properly gets degraded. So you could argue, if you're being formal about it, the reason here why we stay in mitosis is because we never really destroy the kinase. Equally good hypothesis for why we stay in M phase. Well, here we don't have to destroy the kinase because we just turn it off with this powerful stoichiometric protein inhibitor. Pjoink. And now we take the control extract, it comes out after about half an hour, and in the phosphatase depleted extract, it also comes out after half an hour. Shit. We're completely wrong. Ugh. Doesn't delay exit from mitosis at all. So Satoru does another. Sorry, no, wait a moment. This can't be right, he says. So it's because we're using sort of weird ways of going into and out of mitosis, very unphysiological. So what he does is a really heroic thing. He forces the entry into mitosis by adding a bolus of GST-tagged cyclin B, okay? And sure enough, that makes you enter mitosis. And then he removes the GST cyclin B. We were going to do it by affinity chromatography on a glutathione column, but there's so much glutathione in the extract that that doesn't work, so we use an antibody instead. But never mind, you can get rid of it and out you come. Great, so now we test, here we go in, here we come out in the MOC depleted extract, and here we come out in the B55 depleted extract. Well, maybe it's a little teeny-weeny bit slower, but basically not at all, you know. This extract has no trouble getting out of mitosis. So bye-bye hypothesis. Meanwhile, a paper has appeared from Sally Kornbluth's lab that say, once again, it's PP1 that gets you out of mitosis. And sure enough, when you add something called inhibitor 2 of PP1, which again is a heat-stable protein, that indeed it really looks like, much to our annoyance, Sally Kornbluth is actually right. And the reason why we thought she was wrong was because when we did the PP1 depletions that she did, we didn't get the same result. And again, we found this much later, many years later. This was due to an artifact, which was that Satoru's antibody against PP1 reacted with one form of PP1, but it was a minor form of PP1 and not the major. So we thought we would, by this antibody, we depleted all of PP1. But actually, most of the invisible PP1 with which the antibody didn't react was still there. So this question, by the way, is still not fully resolved. So now I gave up running. This is completely fraudulent, this talk, because I haven't had a lab for years and years. So there are some new facts which are jolly interesting, and I follow these and try and make sure they get well publicized. So here I am well publicizing them, I hope. So it turns out a Spanish friend, Marcos Malumbres, who's very good at mouse knockouts and has made a number of very telling knockouts in the cell cycle. He's knocked out Great Wall, and I'm glad to say that the Great Wall negative mouse does not exist. But you can make Great Wall negative mouse embryo fibroblasts, which you can then, because it's got a Cree-Lox excision cassette, get rid of Great Wall from these growing cells. And this is pretty amazing, because I thought Great Wall was really important for controlling the timing of entry into mitosis. But the shocking fact is that these Great Wall knockout MEFs show absolutely no delay or problems in entering mitosis. But chromosome segregation looks terrible. It's rather hard to put your finger exactly on what it is that looks terrible. I mean, I think messy is the right word. It's sort of curious. I mean, you know, sometimes you can't actually quite diagnose what's going wrong. Then, very important, Mike Goldberg and others have been, this has taken an awfully long time to find out who is the enzyme that turns off Great Wall at the end of mitosis. And the evidence is really mounting now. It is, in fact, this enzyme. And also that the enzyme that turns off Ensa phosphatase is also this enzyme. But then this raises, wait a moment, you say, how can a thing turn itself off when it's been inhibited by what, right? I mean, this is a ridiculously circular argument. Well, the answer is that the Ensa phosphatase against phosphoensa is 1/3000th of the rate of a good substrate. So, and Mike calls this inhibition by unfair competition. What it is that little substrate gets in there, the enzyme is sort of trying to get it off, but it can't, so it just takes a very long time. So what this is, I think, I mean, one way of thinking about this is, did you remember I emphasized that after the chromosomes come apart and you start degrading everything, you actually don't want to precipitate cleavage too rapidly because you want to leave time for well, this is a perfect timing mechanism, right? You've stuck an inhibitor which is only going to very slowly turn itself back on again. And because the cyclin has been turned off, because CDKs are what turn on Great Wall, so you've turned off the kinase for Ensa, but the Ensa will take a little while, so it's a switch with a delay timer built into it. And this was very recently and sort of triumphantly confirmed, more or less, by a guy working in Hong Kong called, no, Gothenburg, excuse me, Gothenburg. But he's Chinese. This is all very confusing. He's in Singapore, and Nobu is in Imperial. Anyway, Nobu came to me and described these lovely experiments with having terrible trouble getting published. So they wanted some advice. And I thought it was a wonderful paper, but it had a very bad title and very bad abstract, so no wonder the editors didn't like it. They weren't even sending it out for review, although it's tremendously important. So these guys made a Great Wall knockout mouse oocytes, which you can do by putting Great Wall under and these are infertile female mice, right? And what is amazing, if you take these mouse oocytes, they enter meiosis 1 absolutely normally, and the kinetics of entry are absolutely normal, and there's no doubt about it. But amazingly, but they are infertile, because after that they can't keep the CDK1 levels high, and they slip out. And instead of going, what actually normally happens is that you go through meiosis 1, and then the chromosomes never decondense. You never form a nuclei. You just you get rid of the first polar body, and the chromosomes then reassemble on the second meiotic spindle without going through an interphase, really. But in these cases, what happens is that they can't maintain the CDK1 levels, and meiosis 2 just fails completely. Absolutely amazing. And again, this absolutely proves that Great Wall has nothing to do with the timing of entry into mitosis. So that problem is still up for grabs. So in all of this, I go and consult with my friend Bela, and here he is. And this is absolutely typical Bela-ese. He's Hungarian, and he's a bit sensitive. He's a bit sort of touchy about, if you be careful what you say. But he's terrific, and he has absolutely encyclopedic knowledge of the literature. And the problem, as he points out, is this is what the circuitry looks like, I think. And the problem is that this enzyme here that I've been talking about for the last half hour or so is not only, I mean, it does act on downstream targets, there's no doubt about that, but it looks like PP1 is quantitatively the more important. But it also works on the CDC25 and WEE1 targets. So, you know, it's really, really hard to figure out what would happen if you don't have this. And Baylor's genius is to try and plug all these circuit diagrams to make guesses about the kinetic constants and try and see how the system behaves. And it's one of those cases where usually I sort of understand what he's saying for about half a day after visiting him, and then sort of it all slips away again because I'm really not clever. So the thing to do is what I've really told you is it's a latch with a delay timer built in, and the circuit looks like this. CDK turns on Great Wall turns on Ensa, Ensa inhibits this phosphatase, and we don't know really what are the salient targets of those that effect, but somehow it's essential for staying in mitosis, and if you don't have it, you somehow slip out. And you know, now very recently, these guys here, who are also in Oxford, and Baylor's, I can read, there's Baylor's name there, and Francis Barr is the PI of this lab in Oxford, and they call it the egg timer, which is sort of a joke. E stands for Ensa and G stands for Great Wall, right? And actually, they call this the BEG circuit because B55, anyway, it's an egg timer. And I'm not sure that I fully understand how all this is supposed to work. And I suggest if you're at all interested in this, you try and read this paper. But I must say this paper could have done with some more stringent editing, in my opinion. It's jolly hard to understand. Even the introduction, I felt that I was exhausted after reading the first column, and then the second time I read it, I got halfway down the second column, and then, you know, it's one of those things. And the experiments are very beautiful, but figuring out all this stuff is not easy for somebody like me. So then finally, there are some very interesting things that absolutely don't fit, which is that budding yeast contains a Great Wall homologue. It's called RIM15, and it contains Ensa and ARP19 homologues called IGO1 and IGO2. And apparently these have very little to do with cell cycle regulation, but if you get rid of either RIM15 or delete both IGO1 and IGO2, so you can't inhibit the B55 homologue in budding yeast, and you grow these yeast without glucose, that kills them. A normal yeast would sort of mount some kind of protective response and make lots of trihalos and stuff and be perfectly capable of, you can put them in water and they come back to life years later, as you know if you've ever done any baking. But these guys don't. So it seems to be a module which exists but has been plugged into a different control circuit. So that's one thing. Then nematodes have no obvious Great Wall in their genome, and you can delete the Ensa. So why do they have Ensa if that's the case? It suggests Ensa must do other things as well. But if you delete it, it doesn't seem to have any phenotype. And then in Drosophila, if you have mutants in Ensa, you just have to reduce PP2A activity a bit, and they survive pretty well. So I think David Glover, who wrote a review, has taken together these anomalies are reminders that our understanding of the regulation of protein phosphatases in cell cycle progression is still rudimentary. There's still a lot of work needed to sort all of this out. And it's really, I find it, I'm really, you know, quite obsessed by it, and I'm kind of sorry that we can't do any more experiments. But it's much nicer in some ways when other people do the experiments for you, and you can encourage them and take a delight in their results. But I think the thing I want to conclude with is that, you know, these theories and circuits are all very well, but when you do experiments, one thing that's really taught me, you know when you're really doing good science, because nature constantly comes and sort of bites you in the ankles or punches you on the nose and say, yeah, you're doing pretty well, but that's not quite right, you know. And if you don't sort of, you know, if you don't pay attention to those little things that don't look quite right, you're in serious trouble, and you will make a fool of yourself. So I think so far we haven't actually made a fool of ourselves, but we're very far from unraveling the whole truth. So that's where the story stands, and I'm sorry I've gone on for such a long time, because I always do that. I'm really sorry. Okay.